So, you want to do gel electrophoresis. In this case, we're going to be making an agarose gel, and so we need to measure out the agarose. Gel electrophoresis is a method for separating protein, RNA, or DNA. It has many practical applications in addition to more field-specific ones. In forensic science, this technique can be used to determine heredity, like a paternity test, or to catch a criminal. In more field-specific applications, gel electrophoresis can even be used to help sequence DNA.
Next, we add the agarose to the TBE. The TBE buffer is a neutral pH buffer that facilitates the migration of the DNA which is negatively charged from the cathode, the negative end, to the positive end, the anode.
This solution needs to be microwaved so that it boils and dissolves the agarose. Once the solution has boiled, it can be removed from the microwave and needs to be cooled before proceeding. In the meantime, we need to set up our gel mold to pour the solution into, and this will form the gel as it cools. Once our mold is done, and once the TBE agarose solution has cooled, we can add our next ingredient, ethidium bromide. Ethidium bromide, or ETBr for short, is an intercalating agent, which means it sticks itself into the DNA, and when exposed to ultraviolet light, will fluoresce, and so the DNA can be imaged.
Next, we pour the ETBr, the TBE, and the agarose mixture into the gel casing. We do this and make sure that we minimize all bubbles.
Now it's time to load our DNA sample into the gel. Isolating DNA can be a pretty lengthy process, so we'll save that for another show. Each sample is placed in its own individual well. The pipette must be carefully operated such that the gel is not punctured or the solution spills out into the surrounding liquid.
Here's how the process works. An electrical field is applied through the agarose gel loaded with samples. Because DNA has a negative charge, it will migrate to the positive end of the gel when pushed by negatively charged electrons. Scientists use the aid of tracking dyes in order to see the migration patterns. The agarose gel sorts by size, which means that smaller fragments of DNA, RNA, or protein will be found closer to the positive end of the gel because they are able to move the fastest.
And just like that, your gel electrophoresis is complete. Now you can take your newly run gel and stick it in some imager, and you can analyze those patterns that you get. Join us next week when we'll be discussing how, against every single piece of evidence that's been put out, vaccines really are bad.